We're running just a few minutes behind. No worries. We're running a few minutes behind, but that's fine. We're just going to go a little bit over in the coffee break. We'll start like 10 minutes late and then come back 10 minutes late from the coffee break. So no worries there, I think. Next speaker is Rasmus Byrne, and he's at University of Wisconsin-Madison. He's going to be talking to us about physiology. Thanks.
All right, great. Thank you very much, Tor, for helping organize this, and for all the other organizers and participants here, and for you all to show up this early in the morning. So as Tor mentioned, I want to talk about the basic physiology of fMRI and particular aspects of signal and the noise. I'm just going to begin by a brief overview of fMRI, in particular getting into some of the contrast mechanisms, trying to describe to you what the source of the fMRI signal is, and then also importantly what these various sources of noise are. And in addition to just telling you about the various sources of noise, also present you with the correction methods that are currently being used and being developed to try to address this. And there's three particular parts or sources of noise that I focus on. One is motion, the other is cardiac, and then respiratory effects. And of course there's a lot of other sources of noise, but I think these are some of the more dominant ones. So you just saw a number of really amazing examples of functional MRI data. Here's some from our own lab showing bilateral finger tapping overlaid onto sort of a surface rendering of a structural image of the brain. So the question is, with all these beautiful images, where does that signal come from? How do we generate these kinds of maps where we can overlay just nice function on top of the anatomy? And what we do with MRI is not actually measure the neural activity directly. We'd love to be able to do that. There's some efforts to try to see whether we can do that, but we're not quite there yet. So instead, we're measuring it rather indirectly. So I'm going to walk you through that. So let's say you start performing some sort of task, like tapping the fingers on both your hands. The neurons in the part of the brain that control that movement, say in the primary motor cortex, start becoming active. And as a result of the neurons becoming active, they want more nutrients, and we actually see an increase in the blood flow. Now, it turns out that what gives us the really nice signal is, particularly for the blood oxygenation level dependent functional MRI, or bold fMRI, lies here in the red blood cells. So if you look at the red blood cells there, this was actually discovered quite some time ago, that oxygenated and deoxygenated blood have different magnetic properties. So we have oxygenated blood being diamagnetic, very similar to the surrounding tissues, and deoxygenated blood being somewhat paramagnetic. So if you look at this in the magnetic field, as in the diagram on the right, you see that a bit more of a distortion in the magnetic field around the deoxygenated blood cells. So what happens then is that, say at rest, when you're just lying there, you will see, so this is actually on the right is sort of in a typical resting condition. You have red blood cells coming in through the arteries into the capillaries. In the capillaries, that's where they exchange. They give the oxygen to the tissue, and you see them become deoxygenated, as indicated by the blue circles here, and leaving out into the veins. And the diagram below it, you see are some gradients around the blood vessels that you see, and as a result of that, we see the spins dephasing in sort of an exponential decay. On the left is the case what happens when the area becomes active. And as it turns out, the blood flow increases, and it increases much more than the demand for oxygen. So we actually see a decrease in the number of, or the amount of deoxyhemoglobin there, because it's sort of being pushed out by all that oxygenated blood rushing in. And so in the extreme case, sort of what's shown on the left, is that the gradients kind of balance out, and you basically don't have that dephasing. And as a result, you've got an increase in the MRI signal. So a change in that relaxation curve. So just to recap, what we have is with the task, we see an increase in the task, causes an increase in the blood flow. The blood flow increases more than is actually needed for the metabolic use of the oxygen. And so we see a decrease in the amount of deoxyhemoglobin, and that means decrease in the stuff that is making the signal that is messing up the signal. And so we actually see an increase in the MRI signal as a result. So this is one way, just recapping that again. This is, again, a little bit of a simplistic picture because there's a bit more going on in what we call the hemodynamics. In particular, you also have the metabolic usage of oxygen, the cerebral metabolic rate of oxygen usage, or the CMRO2, increasing, and that's going to create some deoxygenated hemoglobin. In addition, the blood vessels, particularly in veins, dilate as the blood flow increases through them, and that actually leaves more of the deoxyhemoglobin around, so it's not going to leave quite as quickly. So both of these effects, the increase in blood volume and increase in metabolic rate of oxygen, actually counteract against that blood flow. But as it turns out, the blood flow still increases enough to compensate both of these effects, and we still end up with a decrease in deoxyhemoglobin and an increase in the MRI signal. The other point from this figure is, as you see there on the lower left, and you saw some really nice examples of that from Larry's talk, is that the bold response is rather sluggish. So on the middle there is a response shape from a 1 second visual stimulus, and you can see it kind of increasing and then decreasing over the period of 5 to 10 seconds. So this means that this is a pretty sluggish response. We're going to place some sort of limits on the kind of temporal resolution we can image with functional MRI. In addition to that, spatially, as sort of illustrated by that cartoon there, you will see the activation close to the capillaries, but the changes in the oxygenation would also propagate further downstream. And so you may actually see large signal changes further downstream in large veins and draining veins. And so something to be mindful of, that there may be some displacement of the actual activation just based on the physiology. And there's ways people have proposed to try to correct for that. Here's an interesting also effect now in terms of the latency difference. This is actually a slide I got from Peter Banantini. What you see here is the activation amplitude, but also the relative latency from a bilateral finger tapping task. And you can see some heterogeneity in the amplitude of the response within motor cortex, and generally you see the hotspots closer to the larger vessels. But you also see a large spread in the relative latency. And if you notice the scale on this, this is a difference in the latency, and it's just from a very simple finger tapping task within the primary motor cortex of plus minus 2 seconds. And that's a natural variability you'd get just from the variations in what type of blood vessels you're looking at, whether you're looking at capillaries in one voxel or more draining veins in another voxel. It's also a cautionary note that when you want to try to infer timing of neural events based on the hemodynamics, you've got to be very careful because there's this huge spread in latencies just from the hemodynamics. So just because one area of the brain showed a signal increase a second before another doesn't mean that it necessarily was activated earlier. It could just be that you're looking at a capillary instead of a vein. There are ways to get around this to some degree. For example, if you modulate the task, so if instead of, so you perform the task in some way and then you change the task and then you look for how that changed, caused a change in the latency, that is still possible to interpret things that way, to get some information about changes in latency or how things propagate down, because the vasculature stays the same. You're just looking at a sort of a temporal difference. If you're interested in that, I can give you some more examples at the end. So wrapping, as you saw, the typical MRIs that we were getting, this is perhaps a pretty old slide to contrast with what Larry showed in his talk, is that we spent several minutes acquiring a high-resolution image of, for example, this T1 weighted image. And in contrast, for the rapid functional imaging, our goal is really to try to map the changes over time. And so we often sacrifice the spatial resolution to get a more blurry image, but we can acquire that much more rapidly. Now, as I said, of course, technology and new methodology is helping us sort of converge these two, and we can now start to get really high resolution functional or echoplanar images at the similar resolution as the structural images. So it's kind of merging these two disciplines. So we acquired these images throughout time, and during that time we have the subject in the scanner performing some sort of task, such as this bilateral finger tapping, and then we look for where does this signal change in this particular pattern that we had the subject do. So for example, in this case, we had the person tapping their fingers for 20 seconds, resting for 20 seconds, tapping the fingers for 20 seconds, and so on. And then we look for where does the signal change in that kind of a pattern, increasing when they're doing the task and then decreasing again when they're not doing the task. And then we can indicate those with colored areas and then overlay them on a nice rendered image if we want to. The other reason I bring this up is that it highlights another important point in the aspect of fMRI design, and that is how we design the experiment. With fMRI, we're really just looking at the relative changes in activity. We don't get a measure of the absolute neural activity, rather we can just look at the changes in activity. And so it's often a challenge to try to find in a particular paradigm what you're doing, what that control condition should be, or what is the modulation. In this case, it was tapping the fingers versus rest. But in other conditions, rest might not be the appropriate control condition. So for example, if you're interested in studying language function, you could have the person maybe reading or hearing some words and then resting as a condition, but often people sub-vocalize during that resting condition. So another condition may be hearing tones instead of hearing speech, and then you can look at those differences. So it's one of the challenges of designing fMRI experiments is really trying to find out the right kind of control conditions. So now let me get into the second part of the talk, and that is the activation, the sources of noise. And I'm going to cover particularly the ones that you see on the bottom here, the physiological noise, subject motion. There's other sources of noise coming from the scanner, but I'm assuming that you have tweaked all of that and fixed your scanner so that it doesn't provide too many problems there. So the biggest source of noise, I would say, is subject motion. A very characteristic pattern that you might see in your images is positive signal changes on one side and negative signal changes on the opposite side, because that's if your image moves, that's kind of where you see the signal changes. But at certain times, it may not be as obvious as just looking at the time series of images or looking directly at it. You may have to look at the actual time courses, which are shown here on the lower left. So what do we do about that? Well, the best way, of course, is to try to prevent motion in the first place. You can try to pad the head, try to restrict the movement as much as possible. But even with that, we're going to see some sort of motion in some subjects. It's just inevitable in my experience that we see some motion. So the next is to actually perform image registration. And the idea there is, we have an image acquired like this. We basically try to overlap the images and try to rotate and translate those images to try to find the best fit. And then you have to interpolate, of course, to try to get it onto the new grid. And finally, even after doing this step, we often find that we're left with some residual motion signal changes. So a common additional step is to take the realignment parameters that are estimated from this image registration and then include them into the general linear model as additional nuisance covariates and to basically regress out these motion-related signal changes. Now, even with that, you're left with a bit of a problem, and that is if the motion occurs during the volume. So, for example, if I'm collecting a series of slices and the person moves during one of the slices, then if I tried to realign things, these slices would no longer align in a nice stack. So there are some techniques out there to try to do a slice into volume registration. I haven't really seen them used very widely. It's a bit tricky to use them because you also have changes in the spin history effects. One newer direction that I think is really promising and that I've seen some work on in the recent years is what's called prospective motion correction. And the idea here is to actually try to collect additional information that you can then track with the head motion. You can basically collect information about where the subject has moved, feed that back to the scanner, and then have the scanner adjust the slice to track with the motion. And this is actually something that was suggested a number of years ago, probably almost 15 or 20 years ago. But it's only in the recent years we've actually seen some really nice results. I don't have examples from that from the most recent studies, but there's been really nice work from MGH and from others that have been coming out recently. I think some of the challenges with this is, how are you going to acquire that information to feed back to the scanner? You can either collect some navigator pulses from the scanner itself. You can put some probes onto the person's head to try to measure where they are. You can use optical tracking with some camera systems to try to image that. So there's a number of approaches people have proposed to do this. But I think particularly as we move to these more rapid imaging sequences and as the computers are getting much faster, this is becoming much more of a reality. So it's sort of an exciting field to pay attention to. Another very large source of noise is physiological noise, and in particular here I mean cardiac and respiratory noise. So in terms of cardiac noise, we have the blood flow is pulsatile, and so you're going to see some signal changes because of the changes in blood flow. And it's going to happen generally in areas where you see arteries and pulsatile blood, sort of as shown there in the upper right. Breathing, you're going to see it obviously involves movement of the chest wall. And the movement of that chest wall changes the magnetic field at the level of the head. And you can see that actually if you map the magnetic field over time, as shown there in the lower right, you can actually measure these changes in the field exactly coinciding with the breathing changes. And what that's going to do is, remember, a change in the field is going to cause a change in the position. And so you'll actually see a slight shift in the image, very subtle, but a slight shift in the phase encoding direction as a result of that respiration. Now, can you reduce that with just filtering out the data? Collect that. If we say, particularly for resting state, we often filter out anything above 0.1 hertz. And the answer is no, because we have aliasing. So for example, if I had a fluctuation, just a cartoon model here of cardiac signal fluctuating at the cardiac frequency at 1.1 hertz, but if I only sampled that, say, every 2 seconds, which is our typical TR, these are the dots I would end up with. And if I connect those lines, you see it looks like a lower frequency. It's called an aliasing. It's aliased into the lower frequency band. And in fact, it's aliased to a frequency much lower than 0.1 hertz. And so simply filtering out high frequencies at the typical acquisitions that we get is not going to remove cardiac fluctuations. So instead, one technique that's been proposed is retro-ICOR, for retrospective image correction. And the idea here is that we measure the phase of the cardiac cycle and also the phase of the respiratory cycle that each image was acquired in. And then we create sines and cosines of that phase and 2 times that phase, or if we wanted to, we can go even higher. And then we use these additional regressors and fit it to the data. Another way to conceptualize that is what we're essentially doing is we're taking this data. Here's an example time course from one voxel that has very pulsatile blood flow. And let's reshuffle it based on its cardiac phase. We would end up with something that looks like this. And then what we do is we fit a lower ordered sine and cosine Fourier series to that and try to subtract out those variations and try to reduce the fluctuations in time. And you can see that it would dramatically reduce the fluctuations. Now this idea has been around for quite some time. Xiaoping Hu actually proposed the first one of this, retrospective correction, but this was actually done in spatial frequency space and K-space. But done in about 1995, and they showed that you actually get much better localization or much better activation maps when you correct for that. Now, when should you do this physiological noise correction? One of my former students had a paper on this. Tyler Jones, look at that if you want to learn a bit more about this. But it's a little bit tricky because you might think that we're really critical. Depends on the timing of that you want to find out exactly the phase of the cardiac cycle that a particular image was acquired in. So if you move this image and you do the motion correction and volume registration first, you're destroying that information. On the other hand, if you decide to do the physiological noise correction before motion correction, well, if you look at the physiological fluctuation in the presence of motion, you might have a large fluctuation that moves from one voxel into the neighboring voxel. So you see a fluctuation decrease in amplitude and then increase in the neighboring voxel. And so that doesn't really quite fit with the nuisance regressors either. So what we found worked slightly better is actually performing the motion correction first and then doing the physiological noise correction. And I think part of the reason for that is that it's generally motion in a through plane direction that's going to destroy the timing information, and perhaps less so if you're collecting sagittal slices, because most of the motion tends to be head nodding. But it's motion in any 3 dimensions that's going to cause the fluctuations to move from one voxel into the neighboring voxel. And so we found generally doing the motion correction first and then physiological noise correction works better. We actually developed some techniques to try to do both in one step, but it hasn't really shown to be very effective, so still some work to be done there. Another fluctuation, physiological fluctuation that you see is respiration volume changes. So naturally, during rest, your breathing varies. There's periods when you breathe more deeply, periods when you breathe more shallowly, and that in turn causes changes in the carbon dioxide levels. And that's kind of what you see there on the lower left, is just a changes in end-tidal CO2, and which is reflective of arterial CO2, just during rest. It'll be okay. It's one of those auto-advancing ones. So you can see the striking correlation that these changes in respiration volume, these changes in breathing and the changes in arterial CO2 levels have on the signal, and you can see the average signal in the gray matter and the white matter, and you see a really striking correlation, and particularly in those colored areas that you see on the right. So you can also look at that if you look at the just the changes in the envelope of this breathing pattern, sort of looking at the changes in respiration volume. or respiration volume per time, same thing as the minute volume, and map them out, you get very similar looking signal changes. Heart rate is another effect. As your heart rate changes, you may see some blood flow changes, and here's some examples from 2 separate runs where we see that, particularly in the sagittal sinus, you see these kind of changes. And there's been correction methods developed to try to correct for that. Here's one from Katie Chang, RVHR core, when they basically measure the respiration volume, the heart rate, convolve that with some response function, try to fit that to the data, and you see that you can account for significant fractions of the variance explained by respiration and heart rate, up to about 30%, depending on where you look. Just sort of zooming in on that. Another way that people have proposed to correct for that is, instead of just trying to derive it from the physiological signals, sometimes you may not have those, is to average the signal in areas where you don't expect to see function, neural function. So for example, in the white matter or in the ventricles. So you would average the signal in that and use that as nuisance regressors. And as you saw from that first slide, you still, on the average signal in the white matter, still reflect some of these breathing related changes. I should also note that some people have suggested using the global signal to correct for a lot of the noise, sort of taking the brain mask and then averaging the signal of the whole brain and regressing that out. But that has a whole bunch of other problems because that can also include the true neural function. So I'm happy to get into more of that in discussions afterwards. But currently, my lab doesn't use the global signal because of these problems. People have suggested additional corrections, for example, using average signal from the soft tissues, sort of around the head, as an additional nuisance regressor, and in addition also shifting these average white matter and average CSF signals to account for some of the latency differences relative to what you see in gray matter. So for example, this technique, PST core from Jeff Anderson. There's also been ICA-based techniques like the fMRI B's ICA-based X-noisifier, one of my favorite acronyms here, to try to correct for that. The idea is that, and more recently this is now actually more specifically for motion artifacts, but the ICA aroma. The idea here is that you perform an ICA decomposition and then you look for components that are more characteristic of noise as opposed to true signal. The latest one, the ICA aroma, tries to do that in a bit more automated fashion to try to categorize what is signal and what is noise. And if you look through there, there's actually been a large number of techniques that have been proposed and developed over the years to correct for physiological noise and motion as well. And I think that's perhaps one of the challenges in the field right now is that there's so many techniques and trying to figure out which ones are good and which ones aren't. And wonderful acronyms there. We've got Apple Core, we've got Pear Core. I'm just waiting for somebody to come up with Orange Core so we can compare Apple Cores and Orange Cores. But we get accused of comparing apples and oranges. So the other question I want to raise is, should we perform physiological noise correction? I mean, I've shown you here that it does reduce the fluctuations of the heartbeat and respiration, can reduce either false positives or false negatives. It's a bit more important for functional connectivity analyses, where basically you're taking a voxel and saying, okay, what else has this fluctuation in it? And if you happen to grab a voxel that has a large component of physiological noise, you can see all the other areas that have high fluctuations in physiological noise. But you also have to keep in mind that some of these variations in physiology may actually be related to neural activity. Some of my psychiatry colleagues actually use heart rate variability as a behavioral measure. And so I think this is still an open question in the field whether or not you want to apply and would do this physiological noise correction. I think if you're looking at certain areas that say are closer to pulsatile vessels, it makes a lot of sense to do the retro core. But some of these others, looking at heart rate variations and respiration variations, I would be a bit more cautious just because they can, in some cases, correlate with the true function. So finally, just wrapping up one concept, and that is looking at signal to noise. And typically, when we're looking at, there's two ways of defining signal to noise, either spatially or temporally. So spatially, it's basically the average signal divided by the variation in signal over space. And temporally, it's the average signal over time for each voxel divided by the fluctuations of that voxel over time. Now, in a phantom or in an ideal case, you would expect these two to be exactly proportional. As you get better signal to noise, in spatially, you get better coils, for example, or you use bigger voxels, your temporal signal to noise should increase as well. And that's certainly the case if you have purely thermal noise. The problem with physiological noise is that as your coils get better, as you use larger voxels, as you get more sensitive to the actual neural signals you're interested in, you're also more sensitive to the physiological fluctuations. So signal goes up, noise goes up exactly proportionally to that. And so if you're looking at purely physiological noise, you see actually no improvements in the temporal signal to noise as a result of that. So what we see is actually the combination of both of these effects, because we have both physiological noise and thermal noise, and what we see is a sort of red curve here. And so there's regimes where you may be dominated by physiological noise, and then there's certain regimes when you're dominated more by thermal noise. So here's an example actually taken from actual data, mapped out where you collect the data at multiple different flip angles. So at lower flip angles, you have less signal, and it starts to push you to the left on these curves, and you start becoming more thermal noise dominated. You can also notice that the higher curve, this is showing the signal averaged over white matter, and then as you get lower, you see average signal in gray matter, vessels, and CSF. So CSF, in a way, it's like white matter. You see the lowest fluctuation because it has the least physiological noise in it. And so as you get to higher levels of physiological noise, like the gray matter, which contains a lot of the more vasculature, well, more physiological noise, it sort of drives that down, and so you see, reaching a plateau earlier, and similarly in CSF, you see a lot of fluctuations. So something to keep in mind that when you're improving the signal to noise, let's say getting a better coil or getting better voxel sizes, you don't necessarily see that being realized in terms of better detectability in your fMRI. And so instead, what people then do is say, well, might as well use that signal to noise for something advantageous like getting higher resolution images. And so acquiring things at higher spatial resolution, lower spatial SNR, but oftentimes not as much lower temporal SNR. So something to keep in mind. So finally, just wrapping up, I hope I've given you a good sort of overview of the sources of the fMRI signal, but also the various sources of noise and methods to correct for that. And finally, I also want to thank the various people who contributed slides to this and some of the work that was shown here. Thank you very much. Yeah, if there's any quick questions, I'm happy to ask them. Otherwise, we can move to the next. Okay. Good. Thanks.